Okay, we've talked about the establishment of parasegments with engrailed at the anterior margin of every parasegment and stripes of even skipped and Fujitaratsu, which then give us a parasegment. And from this we need to get to segments, and this parasegment will help us with that. And let's just have a look at then how the segments are. So we've taken examples like this before. We have a larval cuticle and it has some segment-specific denticle belts, as they're called, and these are chitinous structures. They are a bit like small hairs and they can be used to sort of grab in the food as the animal crawls along. So here is the anterior, the head, posterior, and in this case we have ventral is on the upside. Okay, so this is its belly, its ventral side. And these hairs can be used to grab in the food as it crawls along and eats. Each segment has these rows of denticles anteriorly in the segment, and then naked in the posterior region of the segment. And then there are, in fact, here, if we look at this, there are 6 different types of rows of denticles. So there's a kind of a anterior posterior polarity even within this segment. And then screening for genes that were important for this, they found that segment polarity or segmentation mutants, they have altered denticle patterns in each segment, and 2 of the genes that were important for this were wingless, the Wnt pathway, and hedgehog, similar to sonic hedgehog. So a wingless mutant will give an anterior region duplicated in mirror image, so the entire segment will be covered in these denticle bands. It will spread all the way down here. And the hedgehog mutant has similar phenotypes. So both of these are clearly important for patterning in the segment. And they could show then that wingless and hedgehog, they know they are secreted signaling proteins that are involved in an intercellular signaling circuit that maintains the parasegment boundary. So a signaling between the cells as opposed to intracellular, okay, intercellular. And they form gradients that contribute to this anterior posterior patterning within the segment. And the other segmentation genes picked out in screens, they encode for components of the wingless or the hedgehog signaling pathways or transcription factors such as engrailed. So if we look at hedgehog now and patched, the receptor for hedgehog, when there is no ligand hedgehog bound to it, acts as a repressor of another cell surface protein, smoothant. And smoothant normally functions to hold cubitus interruptus from translocating to the nucleus, is the classical model. And cubitus interruptus together with suppressor of fused can act as a transcriptional activator. And when we have hedgehog bound to patched, the inhibition of the smoothant receptor is blocked. We get phosphorylation of the smoothant receptor, and this will release cubitus interruptus from the inhibitory complex, where it can then bind with suppressor of fused, translocate into the nucleus, and elicit transcription of the target genes, for example TPP, but interesting for us in this case with segmentation is wingless and engrailed. In the case where no hedgehog is present and smoothant inhibits cubitus interruptus formation or partnering with suppressor of fused, the proteolytic cleavage actually produces a small repressory fragment, cubitus interruptus, the repressor fragment, which can then travel to the nucleus and cause transcriptional repression. So when hedgehog is present, we will get transcription of wingless and engrailed and other target genes. The hedgehog ligand itself undergoes somewhat complicated processing involving cleavage and the addition of a palmitoyl group, which can anchor it to membranes, but there's also diffusible forms of hedgehog proteins that have been identified, and these are believed to generate gradients of hedgehog. So you have some hedgehog that's present on the surface of the cell that can signal just one cell wide to the neighboring cell, and then you can have secreted hedgehog protein that is a diffusible signal that can act over longer distances. That's the important thing to bear in mind. And the other pathway that was important here was wingless, Wnt. And if you remember, there was a non-canonical pathway and a canonical Wnt beta-catenin pathway, which is the important one here. Just to remind you then that in the absence of Wnt, there is a destruction complex composed of APC, glycogen synthase kinase 3, and CK1, an axon, which bind to free beta-catenin in the cytoplasm and phosphorylated and targeted for degradation. Therefore, there is no signaling. But when the Wnt ligand wingless is present, it binds to its receptor frizzled, causing recruitment of a co-receptor, LRP6, and through a sequence of phosphorylation events, we get recruitment of the destruction complex to the Wnt receptor, freeing up beta-catenin and allowing it to translocate into the nucleus, where it can act together with the TCF transcription factor to drive transcription. And important here to keep in mind is that the wingless pathway will also activate engrailed and hedgehog. So already we're seeing that these two important pathways can regulate one of the important compartment regulating genes, engrailed, and that wingless and hedgehog trigger activation of each other's pathways. So this slide isn't on the test, the Capensum. This is a reminder slide, and I also want to expand your knowledge a little bit more in preparation for the next slide that's coming. Just to remind you that we always talk about even skipped and Fujitaratsu as the pair rule genes, but there are in fact 7 pair rule genes, each with 7 stripes. These are shown here. You don't need to know these, but it's just to remind you that they exist and they interact with each other, right? So they repress each other. This is indicated here. They repress each other, and many of them activate engrailed. So what we have when we look at, and here is the nice images showing the expression of them, the different ones comparing 2 at each time. So what we will have is some that can activate engrailed and some that will repress each other. So we get these overlaps, and they're sort of gradients, remember? They're sharp gradients, they're described as. But this will lead to expression of engrailed in a very distinct domain, because some will repress the activators of engrailed and others will act on directly to activate engrailed. Just keep that in mind. So now we come to this slide, which is to help us understand the transition from parasegments to segments by looking at the expression of the segmentation genes in the parasegments and segments. And we'll just go through this part of the figure first. Let's have a look at the color code for players in this diagram. Hedgehog is indicated by these orange cells. They express hedgehog. Wingless is expressed in these green cells here, indicated here. Patched is expressed in these orange cells later on, it's not here. And engrailed expression is denoted by these boxes below the cells. So these 2 cells express engrailed, this 1 cell expresses engrailed. And I just want you to note right away that in this figure, I find it a bit strange. This wingless cell becomes a patched cell, and yes, it starts to express patched, but I want you to keep in mind that it doesn't lose expression of wingless. I find this a bit misleading, this figure, but maybe it's an oversimplification. And maybe I'm going to explain this a bit too thoroughly. It's hard to know. So remember now, up here we have the gradients, the supposedly sharp gradients of the pair rule genes, and even skipped and Fujitaratsu are given. But I showed you this last slide where in fact there are other gradients at play here right? These aren't the only ones. We have peaks of other pair rule genes running through this, okay? And this combination will lead to expression of engrailed here in this cell. The pair rule genes will also induce hedgehog expression. Right? This is written here. And it will be at this stage, 5 hours of embryonic development, it will be such a narrow amount of cells that can interpret this fine signal that it's one cell wide expressing engrailed and hedgehog. Now if we turn to this small inset that I set up here, we described the signaling of hedgehog and wingless and how in fact they can activate each other's transcription factors and how engrailed can activate, for example, hedgehog signaling. So in this cell, the engrailed hedgehog cell, it can produce hedgehog which will activate wingless signaling in the neighboring cell. And hedgehog is actually on the cell surface here, so it can only activate one cell thick and activate wingless in this cell. Why at only this side of the parasegment, right? So here is the parasegment boundary with engrailed in the anterior-most cell and activating wingless in the neighboring parasegment most posterior cell. Why it's only here and not here is presumably due to expression of repressors, the pair rule genes here that will stop wingless expression. For us, the most important thing and what is in the book and could be on the test is that we have a self-perpetuating loop of expression here that is set up, right? So engrailed and hedgehog, you get the secretion of hedgehog to the cell surface. It signals to the neighbor cell, activates the transcription of the wingless pathway, which will trigger wingless signaling, which can then feed back and maintain engrailed and hedgehog signaling in this cell. So this is an important signaling center that is set up around the parasegment boundary. As the embryo develops, it goes through some mitosis, rounds of mitosis in these cells, and we will get then a wider stripe. The embryo is getting bigger, we get a wider stripe, 2 cells thick of engrailed hedgehog here, right? Well, engrailed and hedgehog. And what this cell wants to, this part of the diagram wants to indicate is then that the signaling of hedgehog to the neighboring cells will induce the patched receptor in these flanking cells. As I said, it's still a wingless producing cell, but it also expresses the patched receptor. Meanwhile, the frizzled receptor is expressed in all of these cells, but wingless is just produced here. So this slide just summarizes a little bit what I said, that the parasegment boundary is a signaling center. Here's the parasegment boundary with engrailed expression in the anterior most cells of the parasegment and wingless in the posterior most cell of the parasegment, and that these form a sort of self-perpetuating activation loop signaling between them. Hedgehog to the wingless expressing cell, which produces then wingless and signals back to the hedgehog cell to keep it producing hedgehog. And that's one of the important functions of the parasegment boundary signaling center, is to maintain its own identity in a way. Another important function is that it starts signaling and patterning the segment to be. Okay, so we mentioned that hedgehog in this case was signaling only one cell wide, it was on the membrane, but I said already that hedgehog can be secreted as well, and it's thought that hedgehog will act as a morphogen in the developing segment. Wingless is secreted anyway and spreads sort of far and wide, and this is shown here a little bit. And what we see is that now this is the parasegment line with hedgehog and engrailed here, and the wingless cells in the posterior most of this parasegment and they start creating morphogen gradients. So wingless will be secreted, giving rise to stronger levels of wingless here and weaker levels here. Meanwhile, in the anterior part of the parasegment, hedgehog is secreted, and this is best seen here, and forms a gradient here. Now, if you're like me, you're wondering, why is this gradient just shown in one direction? And that is because hedgehog and wingless have a common target in the EGF receptor signaling pathway that is important for patterning this epidermis. And I have a slide here to show that. The most important part of this slide is here. It shows that hedgehog production will stimulate a protein called rhomboid that is important for EGF signaling, while wingless will suppress it. So since hedgehog is signaling will be strongest around this area and away from the wingless signal, we get the gradient in that direction, right? Because as it moves here, it won't be able to, wingless is closer, the source of wingless signaling is closer and will block the effects of hedgehog indirectly. So that's why the gradient looks like this. I hope that makes sense. Then part of the differentiation process, and not only will it be the production of sort of different structures, these identical belts on the surface, but also we will get the segments forming. And the segments form with the engrailed hedgehog cells in the most posterior part. So engrailed defines the posterior compartment of the segment. And now we have an anterior posterior patterning in each segment. I give this reference where I lifted this out of, and I'll put the paper up on the article or on files on the mol213 folder. This brings us to other areas in the animal that are also patterned during the embryo, and that is the tissues that will give rise to the adult, right? I mentioned these before. They are imaginal discs that are, if we look here at this picture, at one segment, we see that there is a prospective imaginal disc tissue here, and they are placed at these parasegment boundaries, and they will take cues similar to what I've described in the epidermis here, and they will be given posterior and anterior identities based on the positioning here, overlying the patterning that's going on that we just described. And then we mentioned before what a compartment was. It was a small group of founder cells and their descendants that remain within a boundary and never mix with the adjacent cells. And they act together. They usually differentiate in the same way, so they're under common genetic control. And engrailed defines these boundaries of parasegments, segments, and in the imaginal discs, the posterior compartment is defined by engrailed. Here we see a fly with, it could be an engrailed GAL4 driving expression of UAS GFP, and we see its expression in the posterior segments in the abdomen and in the posterior portion of the wing, as well as the posterior parts there of the thorax. Earlier in the lecture, I mentioned genetic mosaics and mitotic recombination experiments where they use these techniques to figure out the role of engrailed setting this compartment boundary. So I thought we would go through this technique, which is a very special and useful technique in Drosophila and very powerful for a certain type of studies. So let's have a look at it. So a few definitions. Genetic mosaic. So we know that embryos are derived from one diploid genome, and typically we would think that all of our cells then are contain the same one diploid genome, but we can have mixtures of cells and some of the cells having rearranged or inactivated genes. And a typical example of this would be a mutation that gives rise to a clone of cells that are cancerous, that generate a tumor, right? You see I already used the word clone there, that they would be a copy of each other, and they would actually be different from the diploid genome. So a human with a tumor could be said in one way to be a genetic mosaic. There's other examples of that, but that's maybe the easiest one to understand. These rearrangements can happen in many ways, but we know that X-ray radiation can induce breaks in DNA, and we can use this as a tool to generate breaks. And when these breaks get repaired, let's say, the easiest way, it's not always that we get the right. So it uses, if one part of the chromosome is broken, it uses the other homologous chromosome as a template to repair the break. And sometimes, with some frequency, there is an exchange of the chromosomes during the repair process at the break point. And this then, when these cells multiply after this exchange of genetic information, we get these fusion chromosomes forming, which can be pretty normal, except that they've exchanged part of the homologous chromosome, and then they give rise to the next cell when it's diploid, having two copies of that region of the chromosome, right? I'll explain this a bit better afterwards. But this is called mitotic recombination. It's not a normal thing. Normally when we speak about recombination, we're thinking of meiosis, meiotic recombination that happens in the germ cells. So here we have in the somatic cells an induced type of recombination that's dependent on this repair machinery, double-strand break repair machinery, that can give mitotic recombination. So we can use this technique to study homozygous mutant clones of cells in a heterozygous background. That's the key use of it. So if you have the normal cell here, you have a mutant chromosome on one chromosome arm, on the other you have the wild type allele. You make blast the chromosomes or the animal with X-rays, and with some frequency you will have breaks at a place that facilitates chromosomal exchange. The breaks can be everywhere else as well, but we won't see them. But if we have a mutant allele or a marker, even better, that we can track visually, a phenotypic marker on cells, that we can track the crossing over event later on, this will make it easier to spot. So then we have a crossing over, this repair machinery, and we get an exchange of information at this crossover place. Sometimes, right? Most of the time it'll give rise to cells that are repaired correctly, but occasionally one of these chromatids will swap with the other chromatid on the homologous chromosome, giving rise to a cell that has one sort of normal looking chromosome with the mutation and one wild type chromosome but with the mutation in that region also present. Instead of normally at mitosis you will get one copy of each chromosome. So you will still be, if this was a recessive mutation, you wouldn't see it, right? But in this case, be it a recessive mutation or not, suddenly it's homozygous for this recessive mutation in this cell, and this cell will go on to expand, and if it had this recessive mutation gave rise to a visible phenotype, for example these multiple hairs in the wing, we would spot this clone of cells then in the adult. We could look in the microscope and see this hairy patch of wing cells. This is conceptually a little bit challenging to understand. I'll go through it in the next slide as well. But so the use of X-rays to make these random breakpoints was how this technique started, but it has been improved over time. And again, the fly has borrowed techniques from yeast here. So yeast has a recombinase called FLIP that recognizes FLIP recognition target sequences, FRT sites. An example of this type of crossing is given here with GFP as a marker. And here we have again X-ray induced mitotic recombination. So you can have a look at this. I want to point out that the FLIP-As can act in other ways as well. If you have two copies of FLIP or FRT sites flanking a gene, you can add FLIP-As, express it via heat shock, for example, or in a certain tissue, and you can then, it causes a recombination that deletes the gene actually. But this can be useful to delete genes, and if you've heard the term Cree-Lox, this is very similar to the Cree-Lox technique. And this experiment is showing how we can use clones to mark regions, and how the engrailed function was demonstrated using this technique. So first they made sort of normal clones, and they see how big the normal clone of cells grows with a marker, for example, multiple wing hairs. If they did this experiment in the minute background and they made a clone of cells here, this clone would, because it was healthier than the minute cells surrounding it, it would take over. And what they saw was that it would take over only within this compartment. It would never cross from the anterior compartment to the posterior compartment of the wing, right? But then if they repeat the same experiment in an engrailed mutant that set the compartment boundary and defined what was posterior cells, then they saw that the clone could overgrow and pass this compartment boundary. So that was how they figured out the function of engrailed, to define posterior and to define the anterior posterior boundary. So this brings us to the last part of this talk, where we meet the homeotic selector genes, which are involved in maintaining segment identity. And this is just a reminder of what we've sort of gone through so far. We have the maternal genes, which set up the gap genes, which then set up the para segments, the pair rule genes, which contribute to the setup of the segmentation gene expression to give rise to these segments and anterior-posterior patterning in each segment. And finally, we come to these homeotic selector genes, and the expression of these genes takes its cues from gap genes and pair rule genes. So the homeotic selector genes, they specify the identity and developmental pathway of a group of cells, for example, in a compartment or in a parasegment. They encode homeodomain transcription factors that control the expression of many other genes, and their expression is required throughout development to maintain identity. There are two complexes in Drosophila of homeotic selector genes that control segment identities. We have the antennapedia complex, or antennapedia C, shown here, and the bithorax complex, BX, the bithorax complex. And if we have mutations in a homeotic gene, they give transformations of whole segments or structures into another related one. So we have a normal one here, and then we have a UB thorax, or is it ultra bithorax mutant, and we have a transformation of a posterior thoracic segment into a more anterior thoracic segment. We mentioned this before, that the halt here is normally, there's normally a halt here in this part that is a primordial wing that has the potential to develop into a wing, and if we have the ultra bithorax mutant, we get this transformation of this entire segment. And then we have antennapedia, which is important for the development of antennae in the head, and mutation of them, the antennae then develop into legs instead. These were cloned in the 80s and found to be related to the Hox complexes in vertebrates. And one of the unusual and interesting thing about these genes is that they have this collinearity. So the 3 to 5 prime order that the genes are organized on the chromosome correlates with the spatial and temporal identity, the temporal order in which they're expressed. So by that I mean that so this gene labial will be expressed before this gene in time and will be expressed more anteriorly than this gene. So and this holds true for the whole set of complexes. So labial will be expressed more anteriorly and before, and all these other genes then will be expressed after it in sequence and in location in the animal, with finally abdominal B being expressed most posteriorly and last. So the homeo box is this stretch of DNA, 180 base pairs long, of conserved sequence that was identified in Antennapedia in 1984. And in other genes, the same sequence is present, right? And it's recognized by a homeodomain transcription factor, and the homeodomain itself is 60 amino acids long protein domain that binds to DNA that has the homeo box. So the bithorax complex genes were first studied by classical genetic experiments done by Edward Lewis, and he received the Nobel Prize for this in 95 together with Nussland Vollard and Eric Wischhaus. And he was able to show that there are 3 bithorax complex genes involved or expressed in overlapping parasegments, where ultrabithorax was expressed in parasegment 5 to 12, abdominal A in parasegment 7 to 13, and abdominal B in parasegment 10 to 14. And it was their combined activities that defined the character of each parasegment. So it wasn't one gene per parasegment, rather the combination of expression of these that define the character of each parasegment. The expression domains in the early embryos are determined by the gap and the pair rule genes, and he could see that null alleles, so where mutations that cause no gene function, so no protein produced or functional protein produced, were embryonic lethal and had transformations of entire parasegments. But weaker alleles, which we call hypomorphs, typically, where had some function was still present and some was lacking, these could survive to the adult stage and had localized homeotic transformations, such as shown here, that the bithorax mutant could give homeotic transformations of the anterior compartment of the haltear. And actually you could combine these weaker alleles and get genetic interactions. So if you took bithorax and PBX, which is a ultrabithorax allele, and you put them into the same animal, the animal would survive but have worse phenotypes, and you have complete transformations of entire segments. This figure here shows this expression of ultrabithorax abdominal A and abdominal B, how they are overlapping, but you'll notice, at least from the figure, that they have a graded expression. So again, kind of like a morphogen gradient, that they were stronger at the anterior for ultrabithorax and abdominal B, with a graded expression as you go more posteriorly, and abdominal B was the opposite, was stronger at the posterior region with a graded expression anteriorly. And mutational analysis are demonstrated here. If they deleted the entire bithorax complex, so ultrabithorax, abdominal A, abdominal B are absent, then they had transformation of 9 parasegments after parasegment 4. Here, the following 9 parasegments were transformed to have the identity of parasegment 4. It's not so easy to see from this diagram, but again they would be looking at how the dentical belts looked, these bristles sort of under the animal, how they looked. And obviously then the tail part was missing. That was the most obvious part, but otherwise they can figure out transformations on the basis of how these hairs are looking. And this gives rise to the idea that the parasegment 4 was formed, became the default state, and parasegment 4 itself was defined by Antennapedia, and then this implies that the expression of the bithorax complexes, these 3 genes, they superimpose new identities on top of this default state. So there's already a pattern laid down by expression of the Antennapedia. It has some sort of gradient itself, presumably. When you don't have then the bithorax complex genes expressed over this, you get that as your default state. You get the parasegment 4 as your default state. And then they could add back the components separately, and if they added back ultra bithorax, which was expressed most anteriorly, they saw that the parasegment 4 would be normal, and then instead of parasegment 5 having parasegment 4 identity, parasegment 5 was normal and parasegment 6, but all of the segments after that were transformed to parasegment 6. So it had overlaid a new default state by expression of ultra bithorax. A new default state was imposed upon the other parasegments. And similarly then, when they added back both ultra bithorax and abdominal A, they could rescue the formation of the 7th, 8th, and 9th parasegments. So this expression of abdominal A would override or suppress ultra bithorax expression, basically. And ultra bithorax expression would suppress the Antennapedia expression further anteriorly. So the conclusion was that the bithorax complex genes act in a combinatorial manner to specify identities. And then finally they put back the most posteriorly expressed genes, abdominal B and abdominal A, and the ultra bithorax was absent, and then they see that they get a normal looking terminal or posterior end, and mostly normal looking sort of mid segments, but then they were missing the earlier segments defined by ultra bithorax. So all of them were necessary to give some specific segments their identity. But there's an interdependence between them. Here I'm just describing the collinearity again. It's interesting. You have this 3 to 5 prime order of the genes, and that correlates with the spatial and temporal order of expression along the anterior-posterior axis. This is also true for the Hox genes in vertebrates, and it is not fully understood, but it's thought to contribute to the mechanism that controls expression along the anterior-posterior axis, so that the first gene has some regulatory functions on the second, and so on. Overexpression of ultra bithorax, which you remember is expressed in most of the parasegments between 4 and 13. If you express it everywhere, it doesn't transform these segments. Remember, it was required for segments 5 and 6 identity, 5 and 6. But if you express it now everywhere in the animal, won't transform everything to 4, 5, and 6. And this gives rise to the idea of posterior dominance, that posterior expressed genes actually suppress the anterior genes that were turned on before them. So there's an order in which they're turned on, and they think that one can be impacting on the expression of the other, turning it on, but they know for sure that the later genes suppress the transcription of the earlier genes. This is called posterior dominance. So now just a few words to wrap this up on bithorax and anapedia complex regulation, a little bit on target genes. So the expression of the pair rule and gap genes in the parasegment does disappear, right? They're turning on the Hox genes, homeotic genes, but their expression disappears. So how is the pattern maintained? And some homeotic selector genes have positive autoregulation. So when they are turned on themselves, they will have a self-perpetuating autoregulatory loop. And then, as I mentioned, also some other genes they can suppress, other Hox genes, they can suppress this posterior dominance, for example. So you can establish and maintain expression patterns. Some examples of this are given here. DFD, deformed promoter, has a deformed protein binding site, so it maintains its own expression, whereas ultrabithorax protein is a repressor of the antennapedia gene and will reduce antennapedia expression posteriorly. But some of the targets of these homeotic genes are actually chromatin regulators. For example, the polychrome group proteins that maintain homeotic genes in a repressed state where they are initially off, and the thrithorax group proteins that maintain expression of homeotic genes that have previously been turned on. So there is interregulation between the homeotic genes where they can enhance their own expression and repress other homeotic genes from being turned on in that position, but they also activate the transcription of genes that will keep those genes repressed where they should be and maintain their own expression where they need to be. And then you can start thinking about, okay, that's how those genes regulate themselves, but what are their downstream targets that actually cause cells to differentiate into a certain morphology in the segment? And the short answer to that is there's probably hundreds of downstream target genes, some of which will be transcription factors and have several targets themselves, and many will be signaling components that perhaps are signaling from cell to cell. So it's very complex. And many of the downstream targets then will influence the cell behavior, including cell cycle, growth, cytoskeleton rearrangements, cell adhesion, cell-cell communication. And all of these things will contribute to the final terminal differentiation and the morphology, how those cells look, right? So it's complicated. But, I mean, you shouldn't be disheartened that we're not going to explore further at this stage. If we look at the number of genes that we've actually gone through, it's a significant amount of genes. We've been talking for the last two lectures, right? We started here with, if we just look at the anterior posterior system with maternally contributed genes and how they regulate then the gap genes. We talked about some of the pair rule genes and the segmentation genes, and then we got down to the selector genes, setting up the final pattern, and that's a lot, you know. And then we also talked about the genes involved in dorsoventral patterning. So we have gone deep enough in a way, right, without starting to look at this new round of transcription factors and their targets. And we're almost finished. I just want to mention that we've talked about this segmentation patterning in the thorax and the abdomen, but there are anterior head segments as well that are not covered by these genes. They don't have these Hox clusters, but they do have homeotic genes that are important in regulating this. And initially these are expressed in this kind of a pattern here. We won't go through it much, but these are referred to as the cephalic gap genes or head gap genes. And some of these are homeotic or homeodomain transcription factors. And fly have about 100 homeobox genes, whereas mammals have about 250. And they're not part of a complex that is expressed in this bi or collinearity fashion. They have more distinct patterns of expression and are sort of used where it's not identical segmenting repeats or very near identical segmenting repeats. So I don't think this is too important for you, but just so we don't skip over how the head and the similar stuff going on in the tail, how these things are patterned. So we started this, or the last lecture, lecture 3, looking at setting up patterning in the early embryo, and we've continued now in a way down to the patterning events here, the late patterning events, and we have skipped basically gastrulation in the fly, but we will go through gastrulation in other animals instead, and the processes underlying gastrulation are conserved. So it doesn't matter which animal we look at in a way. It's good to take up some other examples, right? Yeah, and we're sort of, it might feel a bit disjointed that we're taking certain parts, but we have taken this patterning in one block, you know, and it's kind of okay to take these blocks, even if one is at the early patterning and the other is the late patterning and differentiation, because these things, as I said before, they don't occur in a very defined way. They are overlapping, right? Enough from me. Talk to you later. Bye.